Hi, in this last video on genome engineering, we'll talk about the polymerase chain reaction, or PCR, which is one of the most important methods used in every laboratory, and we'll go a few steps in terms of trying to learn to use it ourselves. So we'll talk about the three basic steps of PCR, which is denaturation, annealing of primers, and extension of strands. We'll talk about amplifying mRNA in addition to DNA by PCR. We're going to talk about picking PCR primers in order so that we can amplify a specific DNA sequence, and then we'll show how PCR is used in identity testing in crime scenes or for paternity cases. So the first step of PCR is heating a sample up to near boiling. This is 95 degrees centigrade, and DNA, the base pairing between the bases, is not strong enough to keep the DNA together at these temperatures, so that a double-stranded piece of DNA will separate into two single strands. So here we have the high temperature and the two single strands. We've got the 5' and the 3' ends, and we, in this tube, as it's going to cool down to 60 degrees now, we've put in a lot of primers. If there were no primers present, these two strands would just go back together, reforming the double-stranded DNA. But in a PCR reaction, we've added short primers, which are complementary to portions of these two sequences, and we put them in great excess so that on average, there's so many more primers that it's going to encounter this strand more quickly than the original strands are going to recombine, because these are smaller, these will diffuse faster, and they're more prevalent. And now we have a situation where a polymerase can synthesize new strands of DNA. And that's exactly what happened. Again, so here in this view, we have double-stranded DNA. We've heated it to separate it. We cool it down. Primers are now able to bind. They have enough binding energy. We'll add DNA polymerase and nucleotides, and we're going to extend from these primers. And where before we had one double-stranded piece of DNA, now we have two double-stranded pieces of DNA. So in one cycle of PCR, we've doubled the amount of DNA present. So this slide is showing that same step, only we're going to do it 3 times. We're going to have heating, we'll have cooling, we'll have annealing of primers, and synthesis. So we have 2 copies. Heat them up again, separate the strands, let it cool, have primers anneal, and extend sequences. Have it do that a 3rd time, and now we've got go through that process, and now we have 8 after 3 cycles. So in this slide, we'll look at the first 3 cycles of PCR. Again, starting with a single double-stranded piece of DNA, heating it up to separating it, then cooling, allowing primers to anneal to the single-stranded DNA, and then extending. Now after 1 cycle, we've got 2 copies of DNA. Heat it up, separate these 2 copies of DNA, let the primers anneal, extend, and now we've got 4 double-stranded copies of DNA. And then after the 3rd cycle, we've got 8 copies of the double-stranded DNA. And typically PCR reactions will go 30 to 35 cycles. 30 cycles will amplify a molecule if you double each time by over 1 billion-fold. So very small amounts of DNA present perhaps at a crime scene can be amplified into macroscopic amounts, which will allow routine analysis on gels or other methods. So for studying cells and their biology, we can PCR amplify either DNA or RNA. DNA is just what we've been showing. Here's a piece of DNA to be cloned. We add primers that will hybridize with the red region and the green region, and eventually we'll get lots of copies of this particular strand of DNA. But for instance, if we want a gene without introns, and here we have an mRNA sequence, as we saw at the end of the second video, that we can use reverse transcriptase to make a molecule of DNA. We can use the poly-T primers that will anneal with the poly-A region of the mRNA, or you can have two gene-specific sequences, and now after you've made the first strand of DNA, then there's going to be one cycle where only one primer will bind. Now you're making the second strand of DNA, and now we can do another 30-35 cycles, and we will have cDNA clones of a gene because this originated from an mRNA molecule. So there's really nothing special about amplifying mRNA as opposed to DNA, except for that first step. It uses reverse transcriptase to make a copy of DNA. So this is often called RT-PCR. Sorry for the lousy writing. RT-PCR. Unfortunately, RT also can stand for real-time PCR. So you'll have to allow for the context to tell you whether you think you're making a copy of mRNA or that you're trying to quantify something by measuring amplification as a function of time. Now, the most important skill for producing a proper PCR reaction is picking primers that will amplify the region of interest. Now, normally primers are about, I would say on average, 23-25 base pairs long. Here I've chosen primers in this example of only 6 bases, but again, normally you will do primers of about 25 base pairs. It just involved too much typing and sequence, and it didn't make things clear to see. We will be practicing this method in class, and you'll be expected to do this on a test as well. So normally when people give you a DNA sequence that they want you to amplify, you're just given one strand with no particular markings, but the convention is always that it's 5' to 3', and you're given a single stranded sequence, but that means that that implies what the other strand is, because each base is going to be complementary, C to G, T to A, etc. And you know the DNA strands are anti-parallel, so you have a 5' end here and a 5' end here. So now to amplify, let's say this piece of DNA that's here in yellow, what you're going to have to do is you're going to have to get primers that cover the ends of the sequence you want to amplify. And you have to synthesize DNA in 5' to 3' region, or sorry, 5' to 3'. So you have to amplify DNA in the 5' to 3' direction, and so that you're going to, for your first primer, just use the first 6 bases. Look at this is the first 6 bases or first 20 bases or whatever of the sequence, because it's 5' to 3', and we're just going to add bases here that's going to be complementary to the other strand. Now even though you're only given this one sequence, when you're trying to pick primers, you'll want to go through this process, write it out in the other orientation. Again, label your 3' and 5', figure out what the complementary sequences are, and then, so you're picking one primer from this strand, and you're picking one primer from that strand, because you want to amplify both strands. And so you're going to pick these bases on the other strand in the opposite direction, but since we don't write sequences 3' and 5', the convention is always 5' to 3'. We're going to flip this one around. This one is CTC, ACC. You can see that here. And this is going to be GAG, GAG, CAG, CAG. So here, for this question, this would be the 2 primers. Now in practice, you won't have to do this, but in practice, it's better, PCR works better when you have 2 primers that have similar physical chemical properties, meaning you want them to anneal at the same temperature. And excellent rules of thumb are you want to pick primers that are 22 to 25 base pairs long, and you want to have at least 13 GCs in those 25 base pairs. And then you'll typically have primers which will anneal at about 65 degrees, which is also good for extension. The last rule of thumb, which is only partially met here in this example, is you generally like to have the 3' ends of primers to be GC rich. That means they're going to bind to the DNA sequence a little more strongly and be a little more efficient at working for the polymerase to extend from them, so that it's sometimes called the GC clamp on the primer ends. But you're not responsible for that in this course. All you're going to need to be able to do is to pick primers that will amplify a particular sequence, write them out in the proper way. And this should be an easy several points on the final exam if you can do that properly. Now, the way that PCR is used to identify individuals is there are certain regions of the genome which have repeat sequences. In these types of repeat sequences, these are usually short sequences, and humans have different numbers of copies of these. They're generally without any type of phenotypic consequence, but it's a characteristic of each of our genomes. And by putting PCR primers, which PCR across the repeat region, that you'll get different lengths that are characteristic of individuals. If you check enough of these repeat loci, you'll get a banding pattern. In this case, the paternal chromosome had a larger number of repeats, the paternal had a smaller number of repeats. So it's a smaller PCR band and a larger PCR band. And if you look at several of these loci, like on this example, where you have 3 different loci and 3 different repeat distances, and you PCR across them, here are, for instance, the 2 blue bands, the 2 green bands, the 2 pink bands. You'll see that on average here we have 3 individuals that have quite different banding patterns, even though some of the bands are in common. Now, if you have a sample that's obtained at a crime scene or in a child, and you can match this banding pattern, we can identify that individual B is the source of the blood that was found at a location, or perhaps is related to the offspring. But in general, this, which could also be done by sequencing, but in general, this is a very fast and easy method to be implemented for the analysis of blood samples. Of course, even as simple as it is, there's still some infamous cases of crime labs doing it poorly, in the O.J. Simpson case, and most recently in the Boston, Massachusetts lab, where samples were DNA results were being faked. So, not a guarantee, but nevertheless, if used properly, can identify people with extremely high accuracies. Okay, that will conclude our discussion on methods of genome engineering. In class, we're going to talk about some of the applications of genome engineering, which are highly controversial in our society. There are no right or wrong answers for this, but it will tackle some of the most sensitive subjects about human reproduction and humanness. I would encourage you all to think for a little bit about what you believe is acceptable or unacceptable uses of genome engineering, and also think about what are the reasons that make you feel that, what are the beliefs that underpin these feelings that you have. Okay, thank you very much for listening.